To a screencast where I will be addressing the light-dependent reactions of photosynthesis. And the goals for this are to be able to identify and describe the reactants, products, locations, and purpose of the light-dependent reactions in photosynthesis. And two here to be explaining, and this is a little more detailed, of course, explain the link between the light, photons, chlorophyll, electron excitation, and water. B, how electron flow leads to products of the light-dependent reactions. C, the significance of non-cyclic and cyclic electron flow. And D here, similarities and differences between non-cyclic and cyclic electron flow and the light-dependent reactions and electron transport, oxidative phosphorylations. So before we leave this slide, I want to point out what I said a lot of there, right? So a lot of like electron excitation, electron flow, electron flow, right? Light-dependent reactions, electron transport chain, oxidative phosphorylation. So you should be seeing sort of a theme here that is familiar to you anyway. So here we go. Light-dependent reactions of photosynthesis are taking place in the chloroplast, specifically in the thylakoid membrane of the chloroplast. So this is where these reactions are taking place, the thylakoid membrane. It's going to involve the thylakoid space as well as the stroma. Right? So we've got stroma and we've got thylakoid space here involved as well as the thylakoid membrane. So we're going to be taking in NADP plus, ADP and inorganic phosphate, as well as water. So these are all reactants. Light is going to be necessary here. This is the light-dependent reactions. So this is going to be happening in the thylakoid membranes. The products coming out of there are going to be ATP, NAD, pH, and oxygen gas here. So this is the light-dependent reactions. So in the thylakoid membrane, from a previous screencast, we saw that there are pigments embedded in that membrane. So again, that's what this block is right here, is the thylakoid membrane. And we see that embedded in there are the pigments. We looked at the pigment chlorophyll, and we talked about how these pigments, not only chlorophyll, but also anthocyanins here and carotenoids down here, are responsible for absorbing photons of light. And so this is what's going on when these chlorophyll molecules or other pigments down here, pigments that contain electrons. These are organic molecules that contain electrons, and you see that we put this on an energy scale over here. So this is sort of free energy. So what's happening is that these chlorophyll molecules or these pigment molecules are going to absorb photons of light, and when they do, electrons get excited to a higher free energy level. Right? So these electrons get excited by the absorption of light by the pigments, and so the pigments are the sources of electrons. These pigments here are the sources of the electrons. Now, if we just had this pigment here, the electron would get excited and then it would fall back down to its ground state if we took the light away. And as that electron falls back down to its ground state, it would be releasing energy in the form of heat. And if this is a fluorescent protein, if this was a fluorescent molecule here, some of that energy that comes back down to its ground state, you'd be releasing light. So this is the premise behind glow-in-the-dark pigments in shirts or stuffed animals or what have you, or anything that glows in the dark. It's absorbing photons of light here, that pigment, and then as the electrons fall back to their ground state, when you take the light away, you put that thing in the dark, and it glows in the dark because it's releasing energy in the form of light. Now, in the case of photosynthesis, these pigments are going to absorb the photons, and electrons are going to be excited, and they don't fall back down to their ground state, yet rather they are captured by by proteins that are in the membrane. So this should sound sort of familiar here, what's going on. So here's the thylakoid membrane. Right? Here's the thylakoid space. This is the stroma on the other side of the membrane. So I've got a membrane separating two spaces here. Here is a group of the pigments in what's something that's called a photosystem. So this is photosystem 2, and then over here is photosystem 1. They are numbered not in order, but they are numbered in the order which they were discovered. So scientists discovered photosystem 1 first, and then they discovered photosystem 2 second. But what we see happening over here in photosystem 2 is that light is going to shine on the chloroplast. Pigments here absorb photons and start exciting electrons. So that's the yellow arrow here. So electrons begin to flow and are then transferred to this thing that's called PQ, and those electrons are then transferred to this cytochrome complex thing here, and then those electrons are transferred to this PC thing here, and then finally they're transferred to this photosystem over here. So let's go back and just review that real quick. Photons of light absorbed by pigments. Those pigments then release electrons. Those electrons begin to flow, right, from one protein complex to another. And, oh, wait a second, what's happening here? As these electrons flow past, look what happens. We pump protons across a membrane. So as electrons flow, this must be downhill. This electron flow is downhill. The electrons got excited by being absorbed, the electrons got excited from the pigments absorbing photons of light, and instead of falling back to their ground state, they then began to flow through these cytochrome complexes over this way. And as those electrons flowed, protons are pumped right here. So what are we doing? We're establishing a gradient, right, of protons inside the thylakoid space. Now, the electrons that were excited from this chlorophyll need to be replaced. They do not come from the photons. Let's be clear about this, that the photons of light are not electrons. Right? The electrons are in the pigment molecules. The electrons get replaced by the photolysis of water. So what's going to happen here is that water is going to be converted into this O2. So we're going to oxidize this water, right, and the electrons get donated to the chlorophyll there in photosystem 2. Why does that chlorophyll need electrons? Because electrons got lost because they were excited, right here. So this process of replacing lost electrons to the chlorophyll in photosystem 2 is called photolysis. Photolysis. And this is why you need to continuously water plants. This is why plants need water to run photosynthesis, is this photolysis step right here. Because if you run out of water, you run out of electrons to replace electrons that were lost from photosystem 2 pigments. So let's continue on here. At the same time, light over here is being absorbed by pigments in photosystem 1. So the same thing is going to happen over here. Light is absorbed by the pigments. Electrons get excited, and those electrons flow, right, ultimately to this enzyme right here. This thing is an enzyme. Okay. And, oh, excuse me. The electrons flowing here end up with this NADP reductase, which then the electrons end up reducing NADP, right, to NADPH. So this is a redox over here. Right? Now, there's no as these electrons flow over here, there's no proton pumping going on. The only proton pumping is over here between photosystem 2 and photosystem 1. Right? Now, as we pump these protons, we make this gradient right here, which we'll talk about in just a second. So let's go back and let me just review this slide here real quick one more time. Light is absorbed by pigments in photosystem 2. Those pigments lose electrons, and those electrons begin to flow toward photosystem 1. The electrons that were lost in the pigment here are replaced by the photolysis of water. The electrons, as they reach photosystem 1, here are replacing the electrons that were lost from the chlorophyll and other pigments in photosystem 1 because they were absorbing light at the same time. The electrons from here then flow, from photosystem 1, then flow to NADP reductase, which are then going to grab those electrons and put them on NADP, right, to reduce NADP to NADPH. This NADPH then goes to the Calvin cycle. So here is one of the products of light-dependent reactions. The other product of the light-dependent reactions deals with this proton gradient right here. So here is the gradient, proton gradient here, and here's an old friend, ATP synthase, and the same thing is going to happen here. So what is this process? Where protons are going to diffuse, facilitate the diffuse through here. This process is, of course, called chemiosmosis, and this is the same process that's going on in respiration, right? Moving protons and then phosphorylating ADP to ATP. So here's then the second product of the light-dependent reactions. So the purpose of light-dependent reactions is to reduce NADP plus to NADPH and to phosphorylate ADP to get ATP. Both this ATP and this NADPH are made out here in the stroma, and both of these then will head to the Calvin cycle, or the light-independent reactions. Here's another way to take a look at that, and I'll use this in class to review. But again, here we're just putting this not in structure in place, but rather on a free energy gradient over here. So there's an opportunity to practice reviewing the details of the light-dependent reaction, see if you can follow that diagram, see if you can summarize what's happening there. There is a blank graphic organizer on energy gradient y-axis, so see if you can figure out what's happening there. What we were just describing was non-cyclic electron flow. Non-cyclic electron flow is electrons flowing from photosystem 2 to photosystem 1 to NADP reductase. But the problem is that we also have a we have a cyclic electron flow. So cyclic electron flow does not include this and does not include this step right here. Rather, it just solely involves photosystem 1. So what's happening is that as photons of light strike, these are photons, strike photosystem 1, pigments release electrons. Electrons from pigments get excited to a higher energy level, and then those electrons will flow to this cytochrome complex here, and as the electrons flow through a cytochrome complex, right, they're going to fall down an energy level back here and are replacing the electrons lost in this photosystem 1. As these electrons flow cyclically, right, this way, spurred on by the absorption of light photons, electron flow is generating a proton gradient, and that proton gradient is the energy used for the chemiosmotic synthesis of ATP. So the cyclic electron flow generates only ATP. It does not generate NADPH. Why? Because the Calvin cycle is an ATP hog. It uses more ATP than NADPH. Right? So we have this non-cyclic electron flow described here, and then we have the cyclic electron flow described here. Both of these are happening at different photosystems all throughout the thylakoid membranes. So here's an opportunity to review non-cyclic and cyclic electron flow. And finally then comparing and contrasting electron transport chain oxidative phosphorylation and what's going on in the light-dependent reactions. So I'll ask you to do this in class as well. And take a look at this diagram and be able to sort of write a description here of what's going on. Again, comparing and contrasting similarities, differences between the two. So you should be able to then identify and describe reactants, products, locations, and the purpose of the light-dependent reaction, as well as explaining all of these, and we'll work on that writing in class. Okay, thanks. Bring your questions.